Welcome to B003, cell division. Every multicellular organism starts as a single cell. All the 37 trillion cells in your body descended from that one fertilized egg through repeated rounds of cell division. Two types of division run the living world: mitosis, which produces identical daughter cells for growth and repair, and meiosis, which produces genetically diverse gametes for sexual reproduction. These processes are fundamental to genetics, development, and evolution. NEET examines them thoroughly every year. Let us make sure you understand them completely. Here is a number that will make you appreciate your own biology. Your body replaces roughly 3.8 million cells every single second. Red blood cells live only 120 days. Skin cells last about 2 to 3 weeks. Intestinal lining cells survive a mere 4 days before being replaced. All of this replacement happens through mitosis. The remarkable thing is the fidelity of the process. DNA polymerase makes errors at a rate of about 1 per billion base pairs, and most of those are caught and corrected. Over your entire lifetime, the same DNA that was in your first cell is faithfully copied and distributed to every new cell. When something goes wrong in this process, when cells divide uncontrollably, we call it cancer. Understanding mitosis is understanding both normal life and its disruption. Before a cell divides, it must prepare thoroughly through the cell cycle. G1 is the first gap phase. The cell grows, makes proteins, and checks whether conditions are right to proceed. The G1 checkpoint is critical for cancer biology. It checks for DNA damage. S phase is when DNA replication occurs. The DNA content doubles from diploid 2N to 4N. G2 is the second gap phase. The cell continues growing and assembles the machinery for division, including the mitotic spindle. Another checkpoint in G2 verifies that DNA replication was complete and accurate. The M phase is mitosis itself, nuclear division followed by cytokinesis. Some cells exit the cycle into G0, a quiescent state. Neurons and muscle cells are largely in G0. Cancer often involves disruption of these checkpoint mechanisms. Mitosis has 4 stages, remembered by PMAT: prophase, metaphase, anaphase, telophase. In prophase, chromosomes condense and become visible under a microscope. The spindle apparatus assembles and the nuclear envelope breaks down. In metaphase, chromosomes align precisely at the middle of the cell, the metaphase plate, with spindle fibers attached to the centromeres of each chromosome. This alignment ensures equal distribution to each daughter cell. In anaphase, the sister chromatids of each chromosome are pulled apart to opposite poles. The cell begins to elongate. In telophase, nuclear envelopes reform around each set of chromosomes and the chromosomes decondense. Cytokinesis, cytoplasm division, happens concurrently or just after. In animal cells, a cleavage furrow pinches the cell. In plant cells, a cell plate forms from the middle outward. Result: 2 identical 2N daughter cells. Meiosis produces gametes and is the mechanism of sexual reproduction. It consists of 2 sequential divisions. Meiosis I is the reductional division. It separates homologous chromosome pairs. The critical event of Prophase I is crossing over, where homologous chromosomes physically exchange segments of DNA. This reshuffling creates new combinations of alleles, genetic recombination, and is one of the main sources of genetic diversity. At the end of Meiosis I, you have 2 haploid cells, but each chromosome in those cells still consists of 2 chromatids. Meiosis II is essentially a mitotic division of those haploid cells, the sister chromatids separate, giving 4 haploid cells total. The fundamental distinction: in Meiosis I, homologous chromosomes separate; in Meiosis II and in mitosis, sister chromatids separate. Let me give you the key numbers and facts. Mitosis, diploid 2N goes to 2N. Meiosis, diploid 2N goes to haploid N. In humans, somatic cells have 46 chromosomes in 23 pairs. Gametes have 23 chromosomes. The DNA content through the cell cycle: at the start of G1 it is 2C, doubles during S phase to 4C, remains 4C through G2 and the start of mitosis, then is back to 2C after division. In meiosis, the DNA content halves twice, to 2C after Meiosis I and to 1C after Meiosis II. Crossing over, technically called homologous recombination, happens during prophase I when the chromosomes are physically interlocked at structures called chiasmata. It occurs between non-sister chromatids of homologous pairs, creating chromosomes with new combinations of parental alleles. Your NEET strategy for cell division. The most tested distinction in this entire topic is what separates in each division type. In Meiosis I, homologous chromosomes separate. In Meiosis II and in mitosis, sister chromatids separate. Questions will give you a diagram or description and ask which stage is shown. Know your stage is cold. Crossing over is another major topic. It occurs only in prophase I of meiosis, specifically when the chromosomes are paired in a tetrad structure called a bivalent. Crossing over does not occur in mitosis. This is a very common distractor. Also be careful about the language. Meiosis II is similar to mitosis in mechanism, but you are starting with haploid cells that already had their homologs separated in Meiosis I. 5 key takeaways from B003. 1. The cell cycle has 4 phases, G1, S, G2, M, with checkpoints that prevent damaged DNA from being replicated. 2. Mitosis uses PMAT and produces 2 identical diploid cells for growth and repair. 3. Meiosis produces 4 haploid genetically distinct gametes for sexual reproduction. 4. The critical distinction: Meiosis I separates homologous chromosomes, Meiosis II and mitosis both separate sister chromatids. 5. Crossing over in prophase I is the key mechanism generating genetic diversity. In B004 we explore DNA itself, its structure, how it is replicated so faithfully, and how mutations arise. See you there.